Welcome to another deep dive.
Yeah.
Today we're going to be looking at something I think is particularly relevant to you as a medical microbiologist.
Oh, absolutely.
And that is underexploited pathways and targets when it comes to developing antibiotics.
Right.
So kind of going off the beaten path
Yeah.
a little bit. And we're going to be taking a look at this through the lens of Walsh and Wensich's book,
Okay.
Antibiotics, Challenges, Mechanisms,
Yeah.
Opportunities, specifically Chapter 18,
Gotcha.
which is called Underexploited Pathways and Targets for Antibiotics.
Okay.
And I think what we're hoping to do today is kind of explore some of these avenues in antibiotic development that, maybe haven't gotten as much attention.
Right.
And try to understand why that is.
Yeah. And it's an interesting question, right? Because we have so many antibiotics that target a limited number of sort of cellular processes.
Right.
And there's this whole world of potential targets that really haven't been completely, explored or exploited. And I think one of the examples they give in the book is fidaxomicin, which is effective against C. difficile infections.
Right.
And it's a really nice example of what a targeted therapy can do.
Exactly.
So yeah, why haven't we seen more success stories like this?
Right. And that's what we're going to be diving into today.
Okay.
So one of the things that they talk about in the chapter is fatty acid synthesis.
Right.
Just disrupting that process,
Ugh.
which seems so fundamental.
Absolutely. It seems like a pretty obvious target, right?
You would think so.
Because it's essential for bacterial survival.
Right.
But the challenge, of course, lies in finding compounds that inhibit those pathways that are essential in bacteria.
Right.
But not toxic to us.
Of course.
And they talk about, isoniazid,
Right.
which is a drug that's been used to treat tuberculosis for many years.
Right.
And it targets anoyl reductase,
Okay.
which is a key enzyme in fatty acid synthesis.
Right.
And then you have some natural products like platensimycin and platensin,
Okay.
which inhibit these fabiacyl ACP syntheses.
Okay.
And then there's also triclosan and derivatives of triclosan that target anoyl reductases,
Okay.
which have actually made it to clinical trials in some cases.
Wow.
So we do have...
So there's been some progress.
Yeah, we do have.
Inhibitors that target those pathways.
But it hasn't exactly revolutionized the field.
Right.
So what's the missing piece there?
Well, one of the issues is that bacteria can be pretty sneaky.
Right.
And so some bacteria can actually scavenge fatty acids from their environment.
Oh, wow.
And so they don't actually have to synthesize them.
Okay.
And so this can be a problem for some of these inhibitors like platensimycin and platensin, because if the bacteria can just get the fatty acids from somewhere else,
They don't need to make it themselves.
they don't need to make it themselves exactly. And so they're not really effective. Okay, so that would be kind of a resistance mechanism.
It is a resistance mechanism, yeah.
Gotcha.
And it seems to be especially prevalent in some gram-positive pathogens.
Okay,
So that's definitely one of the challenges.
that's really interesting.
Yeah.
Now, they also talk about another potential target within fatty acid synthesis, and that's acetyl-CoA carboxylase.
Right. And this is a really interesting one.
Okay.
Acetyl-CoA carboxylase is sort of the gatekeeper
Okay.
of the whole pathway. It's the first committed step
I see.
in fatty acid synthesis.
Gotcha.
And it has these two key subunits,
Okay.
biotin carboxylase and carboxyl transferase.
Okay.
And basically what they do is they activate bicarbonate, and then they transfer that carboxyl group to acetyl-CoA.
Okay.
So it's a two-step process.
Gotcha.
And what's really cool is that there are inhibitors that exist
Oh, wow.
for both of these subunits.
Okay.
So there are these synthetic pyridopyrimidines that target the biotin carboxylase. And then there are natural lipopeptides like moramide and gymramide.
Wow, those are some names.
They are, yeah.
Okay. Well,
And they inhibit the carboxyl transferase.
I see.
And so this really, I think, highlights the potential of this pathway for developing new antibiotics.
Absolutely. And it's interesting that we have both synthetic and natural compounds that are able to target this.
Yeah, it's really exciting.
Yeah, that's really cool. So we've looked at fatty acid synthesis.
Right.
Now let's shift gears a little bit and talk about gram-negative bacteria.
Okay.
Because they have that outer membrane.
Right.
That's really tough to penetrate.
Yeah, it can be a real challenge.
And it's fortified by lipopolysaccharide,
Yeah.
or LPS.
Right, LPS.
So that's a tough nut to crack.
It is a tough nut to crack, but there are ways to kind of chip away at that armor.
Okay.
And one of the ways is to target LPXC,
Okay.
which is an enzyme that's involved in LPS biosynthesis.
Gotcha.
And it's a key enzyme, so if you can knock it out, you can basically prevent
Shut down the whole process.
the bacteria from making LPS.
Right.
And we do have some pretty potent LPXC inhibitors like CHIR090.
Okay.
But as always, bacterial resistance is a significant hurdle.
Of course.
And there's a lot of variability in susceptibility between species.
Okay.
Pseudomonas aeruginosa, for example, is a real problem.
It always is.
It always is. Yeah, it's a tough one.
Yeah.
So, back to the drawing board, maybe?
Right. So we're back to square one with that one.
Well, not necessarily.
Okay.
There have been some modifications to these compounds.
Okay.
For instance, these diacetylenic LPXC inhibitors,
Yes.
they seem to have a broader spectrum of activity.
Okay.
And so they might offer a solution to that resistance problem.
Interesting. So we are making some headway there.
It is promising.
Okay, good.
Okay.
All right. So we've talked about kind of breaching the defenses of
Right.
gram-negative bacteria.
Yeah.
What about just disrupting bacterial multiplication altogether?
Right.
Like, can we target cell division
Yeah, absolutely.
and specifically septum formation?
Right. So the bacterial devisum
Yeah.
is a multi-protein complex
Okay.
that's responsible for orchestrating septum formation.
Gotcha.
And it's really essential for cell division.
Right.
And one of the key components of the divisum is this protein called FTSC.
Okay.
And it's essential for the whole process.
So if we can inhibit FTSC,
Yeah.
we can stop them from dividing.
In theory, yes.
Okay.
But there are a couple of important considerations.
Okay.
One is that FTSC shares some similarities with eukaryotic tubulin,
Right.
which is essential for our own cell division.
Right. Of course, we don't want to mess with that.
We don't want to mess with that, exactly.
Yeah.
So any FTSC inhibitor has to be very selective.
Right.
It has to target the bacterial protein
Right.
and not our own.
Right. And what's the other concern?
The other concern is that, there's the potential for off-target effects
Okay.
on other GTP-binding proteins, which are involved in all sorts of cellular processes.
Right. So we're talking about a pretty delicate balancing act here.
It is a delicate balancing act, yeah.
And they do mention some success stories
They do.
with this.
Yeah, so there's this compound called PC10723,
Okay.
which is a benzamide derivative,
Okay.
and it's been shown to inhibit FTSC.
Oh, wow.
And it's pretty effective.
So it does work.
It does work, yeah.
Okay.
And then there are these other compounds that are even more potent.
Yep.
These improved difluorobenzamide scaffolds.
Okay.
And they've actually shown some really promising results in vivo studies.
Oh, wow.
So, we might be on the verge of actually developing a first-in-class FETSC targeted antibiotic.
That's exciting.
It is exciting.
That would be huge.
It'd be a big deal.
Now, I know we've gone pretty deep
Yeah,
into some of these pathways,
right.
but I have a feeling there's even more
Oh, there's a lot more.
to this underexploited landscape.
Yeah, we've just scratched the surface.
What else is out there?
Well, there are a number of other less explored targets that I think deserve our attention.
Okay.
Riboswitches, for example,
Yeah.
are RNA elements that control gene expression,
Okay.
and they can be targeted by analogs of natural ligands like pyrethymine and rosaflavin.
Okay.
And then there's peptide deformulase,
Okay.
which is an enzyme that's essential for removing the formal group from the initiator methionine in bacterial protein synthesis.
Good.
And it's been successfully inhibited by actinanin and synthetic hydroxymates,
Wow.
although there are some concerns about potential toxicity.
Okay.
And then we can't forget signal peptidases,
Yeah.
which are enzymes that are involved in protein secretion,
Okay.
and inhibitors like arylomycins and actinocarbasins.
Wow.
Show some potential here, especially for synergy with existing antibiotics.
So it sounds like there's a lot more to explore.
Oh, yeah,
A
absolutely.
lot more to this story.
Guns.
Before we move on to that,
Okay.
I want to test your knowledge a little bit.
Mm.
So I've got some exam questions for you.
All right, bring it on.
Are you ready?
I'm ready.
Question number 1. Why are bacterial fatty acid synthesis inhibitors like platensomycin and platensin less effective against certain gram-positive pathogens?
Okay, so this goes back to that resistance mechanism that we were talking about, where some bacteria can actually scavenge fatty acids from their environment,
Okay.
and so they don't actually have to synthesize them.
Right.
And so these inhibitors are just not effective.
Okay, so that's an answer.
That's the answer, yeah.
All right, good. Now, question number 2. Which enzyme acts as the gatekeeper for channeling UDP-GLC-NAC into LPS biosynthesis, and what is a major challenge in targeting it?
Okay, so the enzyme is LTXC.
Okay.
And the major challenge is the variability in susceptibility to inhibitors between different species.
Okay.
Pseudomonas aeruginosa, for example, is notoriously difficult to target.
All right, good. You're 2 for 2. Question number 3. What makes the bacterial divisum such an attractive target for antibiotic development? And what are the 2 main concerns with inhibiting FTSCS?
Okay, so the bacterial divisum is essential for cell division.
Right.
So disrupting it is lethal.
Okay.
The 2 main concerns with inhibiting FTSC are selectivity against eukaryotic tubulin
Right.
and potential off-target effects on other GTP-binding proteins.
Perfect. All right. Okay, here's a slightly more challenging one. Question number 4. Briefly explain how alkyl-depsipeptides, or ADEPs, exert their antibiotic effect on chambered proteases.
Okay, so ADPs act as allosteric activators of chambered protease.
Okay.
And they basically bypass ATP regulation and lead to uncontrolled degradation of essential proteins.
Very good.
Okay.
All right, last but not least, question number 5. What is the rationale for targeting staphyloxanthin production in S. aureus? And what makes this approach particularly promising?
Okay, so staphyloxanthin is a golden pigment that protects S. aureus from reactive oxygen species produced by the immune system.
Okay.
And so inhibiting its production makes the bacteria more vulnerable to our immune defenses.
Gotcha.
And what's particularly exciting about this approach is that potent inhibitors already exist.
Wow.
And they've been deemed safe for use in humans.
Okay. So that's a really promising
It is very promising.
avenue.
Yeah.
Well, you aced our exam.
Right.
Clearly, you've got a good grasp of
Yeah,
these
yeah.
underexploited pathways.
I'll try.
But our deep dive is not over yet.
Okay.
Stay tuned for part 2, where we will explore how bacteria fight back against our antibiotic strategies.
I can't wait.
It's going to be good.
It's going to be good.
All right.
Yeah. It really is a constant kind of back and forth, right? We develop new strategies and the bacteria evolve ways to counter them.
Exactly. And in the last part, we were talking about how some bacteria can actually just absorb fatty acids from their surroundings
Right.
and sidestep our attempts to inhibit their own production.
Exactly.
So it really highlights the need to understand not just the pathways we're targeting, but also the ways that they can get around what we're trying to do.
Yeah, absolutely. And Walsh and Lensvich actually dedicate a good chunk of the chapter to resistance mechanism.
Right.
And they really go into detail about how bacteria fight back against these strategies.
And it's pretty fascinating, some of the stuff they talk about.
It is fascinating.
One of the things that caught my eye was this concept of the SOS response.
Right.
Can you kind of explain what that is?
Yeah, so the SOS response is basically a bacterial survival strategy that kicks in when they're under stress, like when they're exposed to DNA-damaging agents.
So like certain antibiotics?
Yeah, exactly. Certain antibiotics can trigger the SOS response.
Okay, so it's kind of like their fight or flight response.
Yeah, that's a good way to put it.
Okay.
And it's a pretty complex cascade of events.
Okay.
It can temporarily halt cell division.
Okay.
It can repair damaged DNA.
Okay.
And it can even increase mutation rates.
Increase mutation rates. Now, that sounds like it could backfire.
It can. It definitely can, because remember, mutations are the foundation of evolution.
Right.
And so in this context, they can actually accelerate the development of antibiotic resistance.
Oh, so it's like a double-edged sword.
It is a double-edged sword, yeah.
Okay.
So it's a clever survival tactic in the short term,
Right.
but it can also kind of fuel the rise of these resistant strains.
In the long run.
Yeah, exactly.
Now, they mentioned a protein called Lexa
Right.
that plays a key role in all of this.
Yeah, so Lexa is a repressor of SOS genes,
Okay.
and it's also an autoprotease.
So it can break itself down.
It can break itself down, exactly.
Okay.
And so under normal conditions, Lexa basically keeps the SOS response in check.
Okay.
But when the bacteria senses danger, like DNA damage, this protein called REC-A comes into play.
Okay.
And REC-A interacts with Lexa, and it triggers its self-destruction.
So it basically tells Lexa to go take itself out.
Yeah, basically.
Oh, wow. So that allows the SOS response to kick in.
Exactly.
No, yeah, I know the book also talks about the possibility of actually targeting Lexa.
It does.
To kind of disrupt this whole process.
Yeah, and this is a really exciting avenue of research.
Okay.
Because if we can find a way to block Lexa's autoprotease activity, we could potentially prevent the entire SOS response from happening.
So it's like disarming them before they even have a chance to
Exactly.
attempt.
Narrow defense.
It's like taking them out their command center
Yeah.
before they can even issue the orders.
That's amazing.
And there's actually some evidence to suggest that this could be effective.
Okay.
Studies have shown that non-cleavable forms of Lexa,
Right.
basically Lexas that can't self-destruct,
Right.
actually increase bacterial sensitivity to antibiotics.
Oh, wow. So it really does work.
It seems to, yeah.
Okay, but are there any challenges in actually doing this?
There are challenges.
What are we dealing with?
One of the major challenges is finding effective inhibitors.
Lexa's mechanism is kind of unique because it cleaves itself.
Right.
It doesn't cleave another protein.
Right.
And so it's difficult to design traditional enzyme inhibitors
Okay.
that work by competing with the substrate.
So we need a different approach.
We need a different approach, yeah.
Okay, so what are we looking at?
Well, one promising strategy is to design inhibitors that mimic the conformation of Lexa's cleavage site.
Okay.
And so basically you're tricking Lexa into binding to the inhibitor instead of itself.
That's like a decoy.
Yeah, it's a decoy.
Okay, I like that.
And there's been some recent research that's really shed light on the structural features of Lexa that are critical for its autoprotease activity.
Okay.
And so this is really paving the way for more targeted inhibitor design.
So we're getting closer.
We're getting closer.
Yeah, that's good.
It's exciting.
It's very exciting. Now, I know we've talked a lot about kind of disrupting these essential processes, but Walsh and Vensvitz also talk about this really intriguing concept of targeting bacterial virulence factors.
Yeah.
So basically the things that they use to cause disease in the first place.
Right. So instead of killing the bacteria, you're just disarming them.
You're taking away their weapons.
Exactly.
I like that.
It's a really fascinating kind of shift in perspective.
Yeah. Can you elaborate on that a little bit? Like, what are some of the key virulence factors that bacteria use?
Well, it depends on the specific bacteria and the type of infection.
Of course.
But some common culprits include toxins, adhesins, and enzymes that modify host proteins.
Okay, so toxins I think are fairly straightforward.
Right.
They basically disrupt normal cellular processes.
Right.
But what about adhesins?
So adhesins are basically like bacterial Velcro.
Okay.
They allow bacteria to attach to host cells.
Okay.
And this is often a really crucial first step in establishing an infection.
Right.
For example, E. coli strains that are responsible for urinary tract infections.
Right.
They use adhesins to attach to the lining of the bladder.
Okay, so it's how they get a foothold.
Exactly.
Okay, and what about those enzymes that modify host proteins?
So these are kind of like saboteurs.
Okay.
They disrupt normal cellular functions by altering or breaking down host proteins.
Gotcha.
Some bacteria produce proteases, for example, that can degrade collagen, which is a key structural protein in our tissues.
Right.
And so this contributes to the tissue damage that you often see in bacterial infections.
So it's a multi-pronged attack.
It is, yeah.
But the chapter suggests that we might actually be able to block the production or the activity of these virulence factors.
And that's where the idea of antivirulence therapy comes in.
Okay.
And the goal is to basically disarm the bacteria, render them less harmful, and give our immune system an advantage.
I like that.
Yeah.
So how would this work in practice?
Well, there are a few different strategies. One approach is to target the secretion systems that bacteria use to transport virulence factors outside of the cell.
Okay, so you're basically intercepting their weapons delivery system.
Exactly. And there's been some exciting progress in this area.
Okay, like what?
For instance, there's this small molecule called virstatin. And it's been shown to reduce the virulence of Vibrio cholerae.
Wow.
By interfering with cholera toxin secretion.
So we're talking about like disrupting their communication lines.
Yeah.
What are some other strategies?
Another promising avenue involves interfering with quorum sensing.
Quorum sensing, that sounds familiar.
Yeah, so quorum sensing is basically a bacterial communication system that allows them to coordinate their behavior based on population density.
So they can sense how many of their buddies are around.
Exactly.
Okay.
And this can include, switching from individual to collective behavior, forming biofilms, and regulating virulence factor production.
So if we can disrupt quorum sensing, we can basically disrupt their social gatherings.
Yeah, exactly.
Like that.
And there are several ways to do this. We can block the production of quorum-sensing molecules.
Okay.
We can interfere with their ability to bind to receptors.
Yeah.
We can even degrade them.
Okay, so we can jam their signals.
Exactly.
And what about targeting the virulence factors themselves?
That's another possibility.
Okay.
And there are some promising examples.
Okay.
We talked about staphyloxanthin earlier.
Right.
The golden pigment that protects Staphylococcus aureus from our immune system.
Right.
Researchers have actually developed potent inhibitors of staphyloxanthin biosynthesis.
Okay.
And they've shown some promising results in animal models of infection.
So we can basically strip away their armor.
Yeah, exactly.
It's amazing.
And what's particularly exciting about this approach is that these inhibitors have already been deemed safe for use in humans. So this could potentially speed up their development as antivirulence drugs.
That's incredible. So it sounds like we have a whole arsenal
We do.
of antivirulence strategies at our disposal.
Yeah, it's a really exciting area of research.
But are there any downsides or challenges?
There are always challenges.
Of course. What are we up against?
One challenge is that virulence factors can vary dramatically between different bacterial species
Right.
and even different strains of the same species.
So a one-size-fits-all approach isn't going to work.
It's not going to work, no.
Okay.
And so antivirulence therapies might have a narrower spectrum of activity compared to traditional antibiotics.
So we need to be more targeted in our approach.
We do, yeah. And this highlights the importance of rapid diagnostic tools,
Right.
to quickly identify the culprit.
So we know what we're dealing with.
Exactly. So
Right.
we can select the most effective antivirulence strategy.
That makes sense. Are there any other challenges?
One concern is that, bacteria could potentially evolve resistance
Right. Of course.
to antivirulence therapies.
It's the evolutionary arms race
It is, yeah,
that never ends.
unfortunately.
But is there a silver lining to this?
There might be a silver lining.
Okay.
So antivirulence therapies, by their very nature, might actually exert less selective pressure for resistance
Okay.
compared to traditional antibiotics.
How so?
Well, traditional antibiotics usually aim to kill bacteria directly.
Right.
And so this puts immense pressure on them
Right.
to evolve resistance mechanisms just to survive.
To live.
Yeah, exactly.
Right.
Antivirulence therapies, on the other hand,
Right.
aim to disarm bacteria,
Okay.
reduce their threat level
Right.
without necessarily killing them.
So it's less of a scorched earth approach.
Exactly.
Okay.
And this could potentially slow down the development of resistance
Okay.
because there's less pressure on the bacteria to evolve ways to survive.
That's fascinating.
It is. It's a really interesting kind of nuance.
Yeah, so while there are certainly challenges to overcome, the potential of antivirulence therapies is pretty undeniable.
It is. It's a really exciting field.
It is. Yeah, it's a much more elegant approach, isn't it?
It is.
It's like, disarming a bomb instead of just trying to contain the explosion.
That's a great analogy.
And I think this shift in thinking
Right.
is reflected in, the growing interest in these underexploited pathways and targets.
Absolutely.
We're moving beyond the search for a magic bullet,
Right.
antibiotic, and starting to appreciate the subtleties of bacterial physiology
Yeah.
and the potential of these more targeted approaches.
Yeah. And I think it's especially important now, given the growing concern about the impact of broad-spectrum antibiotics on our microbiome.
Right.
The microbiome is this vast community of bacteria that lives in and on us.
Right.
And it plays such a crucial role in our overall health.
Absolutely. Digestion, immunity, even mental well-being.
Exactly.
All connected.
It's all connected and using broad-spectrum antibiotics indiscriminately can really disrupt this delicate ecosystem.
Right.
And cause all sorts of problems.
It's like collateral damage in our fight against infection.
Exactly.
And that's another reason why I think these underexploited pathways hold so much promise.
Right.
Because we can really hone in on specific processes
Right.
that are essential for bacterial survival or virulence.
Right.
And minimize the impact on the microbiome
Exactly.
while still effectively tackling the infection.
It's a much more targeted approach.
Much more targeted, less disruptive.
Yeah.
But are there any drawbacks to this strategy?
Well, there are always drawbacks.
Of course.
One challenge is that some of these alternative targets
Right.
might not be as druggable.
Druggable, what does that mean?
So druggability refers to how easy it is to design a drug that can effectively bind to and inhibit a particular target.
Okay.
Some proteins have very defined binding pockets
Okay.
that make it easy for drugs to latch onto,
Right.
but others are much more challenging.
So it's like finding the right key for the right lock.
Exactly. And sometimes the lock is incredibly complex.
Right.
And it's very difficult to design drugs
Right.
that are both potent and selective.
Right. We don't want them to, interfere with our own cellular machinery.
Exactly. We want to make sure they hit the bacterial target
Right.
and not our own cells.
Now, they also mentioned that some of these pathways might only be essential for bacterial survival under specific conditions.
Right. That's another layer of complexity.
Okay.
Remember we were talking about bacteria scavenging fatty acids from their environment.
Right.
That's a prime example.
Okay.
Inhibitors of fatty acid synthesis might only be effective in environments where those fatty acids are scarce.
Right. So we need to understand not just the pathway itself,
Right.
but the context.
The context,
Which it operates.
Yeah. And knowing when and where a pathway is critical for bacterial survival or virulence
Right.
is really essential for developing effective therapies.
It sounds like a fascinating but incredibly complex puzzle.
It is a fascinating puzzle.
But the potential payoff is huge.
It is.
I mean, if we can successfully harness these underexploited pathways,
Right.
we could really unlock a whole new generation of antibiotics.
Absolutely.
More targeted, less disruptive to our microbiome,
Right.
and potentially less prone to resistance.
It's a future worth striving for.
It is. And on that note of optimism,
Okay.
do you have a final poetic thought
I do.
to leave our listeners with today?
Okay, so this is a reflection on the tenacity of bacteria and the constant need for innovation in our pursuit of new therapies. They are ancient, ever-evolving, a symphony of life revolving.
Huh.
In every niche, they persist and thrive to challenges they constantly strive. We seek new ways with minds alight to meet their strength with equal might. In underexplored realms, the answers reside for a healthier future side by side.
Beautiful. A beautiful reminder that this is an ongoing journey and we need to keep pushing the boundaries of scientific discovery to stay ahead of these remarkable microorganisms. Thank you for joining us on this deep dive into the world of underexploited antibiotic pathways. We hope you've gained some new insights into the fascinating complexities of bacterial survival and the exciting potential of these less traveled roads in our search for new and improved therapies. Until next time, keep exploring, keep questioning, and keep learning.